Awesome. So welcome everyone to another episode of D4P, Data for the People. And this is our COVID-19 series. And so our goal is to provide the science for all straight from the source. And today I'm going to be presenting a paper that talks about the human antibody responses to SARS-CoV-2. Before we get started, I do want to take a moment of silence in lieu of George Floyd, who lost his life unfortunately to cop brutality. As a Black scientist, it would behoove me not to take the time out to recognize that this is still happening. And it's really hard a lot of times for us as minorities to separate ourselves from our professions. And so I just want us to take about 10 seconds just to remember his life. Awesome. Thank you guys. There's so much more I would like to say about this, but I think we'll go straight into the science. So once again, for those who don't know, I am always make a big stink about where I'm from. So I'm actually from the United States Virgin Islands, so our US territory. More specifically, I'm from the large island of St. Croix. We're actually only 84 square miles, about 60,000 in population. And once again, I'm just gonna showcase one of our beautiful beaches. And the weather today, as you can see, I'm in my backyard, but I would much rather it be a beach background. Here's my little family. My husband is on vacation, so he's actually watching our son. And this is him helping me with my presentation, which actually almost went haywire this morning when he spilled coffee on my laptop. But that's a story for another day. All is well, so we'll keep it moving. All right, so a little bit about the publications that scientists put out. So publications is basically a central source where we can communicate our research findings, and it happens in a variety of ways. The problem is a lot of these reports are topic specific and very jargon specific. And so it's really hard for a lot of people outside of the field to kind of depict exactly what the conversation is on or what the data is showing. And so the paper I'm presenting today is actually one from a peer-reviewed article. And so this article is Nature of Medicine. And what peer-reviewed means is that it's evaluated by other scientists in the field, and it's seen as a report that is fleshed out properly, all the data sets look fine, and then it's published. So we talk about, through all the D4P presentations, about the styles of scientific reasoning. This paper looks into experimental evaluation, and so that's using investigations to establish different patterns, to look for differences, or to develop hypothetical model. So for example, in this paper, we're looking at the timing of antibody responses in patient samples. So here's a screenshot of the paper. So like I mentioned, it's from Nature Medicine. And this paper is entitled Antibody Responses to SARS-CoV-2, which is the virus that gives you the disease COVID-19 in patients. Now, what's really fascinating about this paper is that they're not just one nor 10, there are actually about 50 authors on this paper, just highlighting again the collaborative effort that it takes for scientists to put together data sets that can be produced in a platform what can be used to initiate different public health initiatives. Now, throughout my presentation, I'm going to talk about a number of key terms. So one of those terms I'm going to mention is antibodies and antigen. I'm also going to talk about seroconversion, as well as aggregate and individual data points. And so just a brief background on how the body elicits an immune response to SARS-CoV-2. So the virus SARS-CoV-2, as seen here, it's an RNA virus, which you see in the center, and it's protected by a lipid bilayer. Within that lipid bilayer, there are a number of different proteins, but there are also these surface proteins. In this case, it's spike protein that not only elicits an immune response to the virus, but it's also necessary for binding to the receptor on your cell in order for entry and replication to occur. Now, once the virus binds the cell and enters and replicate, it can cause an immune response. So not only is an immune response initiated towards the surface proteins, but an immune response can be made from the proteins that protect the RNA, like nuclear proteins, or other proteins within the matrix of the virus. Now, our immune system is very complex, and I'm only going to talk about one part of the immune system, which is adaptive immunity. But there's also innate immunity, which is like the first line of defense. It's non-specific. It just targets and tries to eliminate anything that's foreign inside of the body. Unlike innate immunity, adaptive immunity recognizes specific antigens. So what occurs, we have a virus, which is a pathogen. So in this case, we have SARS-CoV-2. SARS-CoV-2 enters our cells, replicates. Our body recognizes it as something foreign. And so that foreign antigen triggers an immune response within your body. And so that immune response can go on to do several things. And so the immune response basically recognizes this pathogen and recognizes certain parts of the pathogen, which we call antigens. And so you can either elicit an immune response where different cells have certain receptors on there to bring about many other immune cells to combat infection, or we can make these specialized molecules called antibodies that is used to protect us in the future. Now, these antibodies are fairly specific, so they recognize specific parts of the antigen where they can bind and eliminate infection. And so the paper that I'm going to talk about today focuses on this part of the immune response, which is the production of antibodies that's specific to SARS-CoV-2. So how does the antibody antigen work? And so within adaptive immunity, you have a number of different antibodies that are produced by the human body. There are actually 5 different types of antibodies. You have IgM, IgG, IgM, IgE, and IgA. For our purposes today, I'm only going to focus on 2 antibodies that I'm going to talk about in the next slide. But these antibodies recognizes different parts of the virus. So here's SARS-CoV-2 after it enters the cell. And so these antibodies that are produced can then bind specific parts of the virus, and in this case, the spike protein, and work to either recruit other immune cells to get rid of infection or neutralize the virus so that it doesn't infect your cells. So what are these 2 major antibodies that seems to be implicated in SARS-CoV-2 infection? So we have IgM. So IgM is one of the first antibodies to come into play. Your body encounters a foreign antigen or foreign particle. It recognizes that it does not belong there. And so IgM actually exists off of the cell. So if it's not on a cell as a receptor, it exists alone as a pentamer. So a pentamer is just meaning 5 molecules linked together. And once these molecules enter the bloodstream, it sends messages to the rest of the immune system to produce other antibodies. And in this case, it usually helps to produce IgG antibodies. Now, IgG antibodies are the most abundant in antibodies where you can detect in humans' blood samples within the serum. And so IgG exists as a monomer, meaning 1 molecule. All right, so our first poll, which antibody type is thought to provoke long-term immunity in humans to COVID-19? IgM, IgG, IgD, or IgE? Give you a couple seconds to answer. So 5 more seconds. Awesome, so let's see what you thought. All right, so 48% thought it was IgG. Coming into a close second is IgM with about 36% of the vote. So the majority actually went. And so it's actually thought that IgG is the antibody that is thought to provide immunity for a next infection to that same virus. All right, so one of those key terms I'm going to talk about right now, which is seroconversion. What is seroconversion? So seroconversion is that time point where you go from being undetectable for a specific antibody to being able to detect that antibody within that patient's blood sample. And so here on this graph, we have on the y-axis, we have the time course of infection, and then on the left, we have the amount present. So usually it takes some time from being infected for our bodies to seroconvert to producing a certain amount of antibodies. So you can say at this time point, this patient seroconverted IgM antibodies, and at this time point, they seroconverted IgG antibodies. And I'll talk a lot more about seroconversion in the presentation. Now, another thing that this study points out is the different types of data sets that we have. And so we can either look at data sets as aggregate data. And so this involves many different data points. It can show trends and comparisons using statistical analyses, but it doesn't look at the individual. And so that's where individual data comes into play. And so sometimes when we're looking at aggregate data sets, there might be individuals that may respond slightly differently. And so a lot of times scientists zone into the individual to try to figure out how we can develop personalized approaches to that person's response. So what's the question, right? What did this paper go in to describe and try to answer? And so there's a lot of, there's not much information on there on exactly how our antibodies respond to COVID-19 or how the immune system responds to COVID-19. And so this is a graph, I've also used this the last time when I was describing the tests that we use to develop antibodies. And so there's still some questions that we're trying to understand. So one of the first goals that authors look into is when do we see seroconversion and how long does it last? So when can we see or when can we detect IgM antibodies in a patient's sample? When can we detect IgG antibodies in a patient sample? How long does the presence of IgM occurs? And also, how long does IgG stay after someone is infected with COVID-19? But the authors also wanted to look at something else. So the authors also try to decide if antibody testing can complement viral testing. And so I briefly mentioned it, but in the beginning, you can actually use a specific test called RT-PCR. And I won't describe it now, but if you want, you can go back to Fran's D4P presentation, where she goes into detail as to how scientists can take the genetic information, change it into a way that we can detect it in a patient. And so we use RT-PCR to detect if there are any viral samples or viral particles within that patient who may be currently infected. Another test that we use is antibody testing, such as ELISA, which I've mentioned it in the first D4P talk, so feel free to also check that out, where we look at the presence or absence of either of these antibodies. So does someone have IgM antibodies? Do they have IgG antibodies? Have they recovered from infection, or are they currently infected with SARS-CoV-2? So for this study, I'm going to describe the patient design or how they were able to design the study with the patients that they had. So this study incorporated 285 patients from Chongqing, China, and there were 3 hospitals that they were able to get patients from. Out of these 285 patients, 250 of them had some type of epidemiological history. So what that means is that these patients either were residents of Wuhan, recently have been to Wuhan, or have come into close contact with a resident of Wuhan or someone who have confirmed COVID-19. Out of those 285 patients, 262 of them had clear symptom onset. So what that means is that these patients knew exactly when their symptoms began. So they were hospitalized, the doctor asked them their history, and they could have told the doctor, I experienced fatigue or headache or high fever at a certain day. And out of that 285 patient data set, we have 70 of them where the scientists were able to gather multiple serum samples to look at the seroconversion that I'm going to talk about later in the presentation. So one of the first questions that the scientists ask are, when are antibodies produced after someone's infected with SARS-CoV-2? And so scientists are looking at the presence of these antibodies that they're able to detect after these patients started having symptoms. So what we're looking at is when patients are um, producing IgM antibodies and when they're producing IgG antibodies, and how long after their symptoms are these antibodies being produced. So to answer this question, they used 262 patients. Of these patients, they had 363 serum samples. And so serum is just part of the blood that we can detect the antibodies. And so they have 363 because they have multiple samples from certain patients that I described before. Now, from these samples, they use a type of ELISA to detect the presence of the IgG or IgM antibodies. And so what do they see? So here in this graph, I want to highlight a couple of things. So let's zone in on the x-axis. And so here we see the days after the patient started having symptoms. So we have data set from about 2 to 4 days post having symptoms to about 23 days after they started having symptoms. On the y-axis, what we're looking at is the positive rate. So how many of these patients are we able to see that we can detect either IgM, IgG, or both of these antibodies? So we're not looking at the amount of antibodies, but just if they can detect it or if they cannot detect it. And so what we're seeing at about 20 days after patients have symptoms, you have about 94% of them where you can detect the IgM antibodies. And at about 17 days post symptoms onset, 100% of the patient samples, they were able to detect IgG antibodies. But what this chart doesn't show us is the amount of antibodies present within these patients that have contracted COVID-19. And so next, the scientists wanted to see what the levels were of these antibodies within these patients. So for this data set, we are using 262 patients. And so I'm gonna show you the first data set that they show, where on the bottom of the x-axis, N is the number of patients that they're assessing under each time point. Above the N number, you have the days after symptoms onset. So here we have within the first week, within the second week, so on and so forth. On the y-axis, we're looking at the levels of antibodies. And just a reminder that this is a log scale, so it's exponentially more as you go up the scale. And so the authors also do statistical analysis to show a number of things. So I also want to point out that this is an example of aggregate data that includes individual data points. So all of these dots are the individual patients, but we're using statistics to look at the median or the mean of all of the data points together, so we can look at the aggregate data or the data on a whole. And so this box indicates the first and third percentile of patients. The bar within the middle of the box look at the average of all of those patients together. And then we have the whiskers. So anything outside of the whiskers, we can say that these patients were outliers within that sample set. So this is IgG that we're looking at. And what you can see if we're looking at the averages of all of the data is that you see a continuous increase in the levels of IgG antibodies up to about past 3 weeks post symptoms onset. So the authors also looked at the amount of IgM antibodies levels in the patients. And so what you see here is on the bottom, we have the number of patients that they're looking at and the days after they started having symptoms. And so here you can see as well, when we're looking at IgG, the averages of this aggregate data, you see a trend of an increase of IgM antibodies in terms of the levels that we're seeing. And after 3 weeks, you start to see a slight decline of that levels. Once again, there are some individual data points that are outliers, but in order to get a picture of the population as a whole, we have to use this aggregate data set to look at the averages so we can infer how the population reacts to a certain stimulus. So overall, this slide is showing that we're seeing a slight decrease in IgM after 3 weeks. So what does this change what we know? So in the first graph that I've been presenting, I'm showing that initially you're seeing IgM antibodies being detected, and then you're seeing IgG antibodies detected with IgM decreasing as IgG increases. Now this changes the graph a little. So what we're starting to see is that it seems that antibodies are produced around the same time. And after about 3 weeks or so, you're seeing a slight decrease in antibody levels for IgM, but you're seeing a continuous increase in IgG. And the authors also point out that some of the patient samples started to remain steady or plateau throughout the time point. Now, there's a lot of talk about whether or not a patient who has a more severe case of COVID-19, if they produce more antibodies than someone who has a less severe case of COVID-19 or are asymptomatic to infection. And so what the authors do to combat this question is that they look at a subset of patients who they classified as severe. So severe would be someone who's been admitted into the ICU. And so out of the patient cohort, they had about 20 that they classified as having severe. So the orange dots are severe cases, the green are non-severe cases. And you're also looking at the time after the patient started having symptoms. So here in this portion of the graph, we're looking at the first week after patients started to have symptoms. And here we're looking at the second week after they started to have symptoms. Now, this data set shows, we're gonna zone in on the median of this entire patient data set. And we're seeing that there seems to be a trend of people who have severe cases of COVID-19 have higher levels of IgG antibodies being produced, unlike those who have non-severe cases. But I also want to point out, if we were to zone in on the individual data, that we do have some patients who are also producing large amounts of antibodies, and even though they don't have severe symptoms. They looked at the same thing at post 2 weeks of symptoms onset, and you do see a slight increase of antibodies produced in comparison to those who have non-severe. The only significant difference that they show is within that first week with IgG. And so what the p-values indicate is how much that this phenomenon is occurring by chance. And scientists pick a number which is p equals 0.05. And if that number is less than that, we can say that the chances of that happening by random chance is very low, and it actually does have something to do with the phenomenon that they're looking at. The authors also looked at IgM. And so here, again, if we're looking at the median for each of the data sets, we see within the first week, you do see a slight increase of the amount of antibodies produced for severe patients. And the same is true for those post 2 weeks of symptoms onset. Once again, this is a small data set, so we can only come up with conclusions based on the sample size that we have. So what did the authors show so far? So all of the patient samples that they've tested, so looking at their serums and looking for detectable antibodies, 100% of them produce IgG antibodies about 2 to 3 weeks after they started to have symptoms. IgM antibodies, the amount seems to decline after about 3 weeks of having symptoms. And there seems to be a trend where patients with severe symptoms are producing slightly higher levels of IgM and IgG, with significance only seen in the first week post-infection with IgG antibody levels. All right, so I'm gonna dive in a little bit deeper on the seroconversion of antibodies. So I mentioned seroconversion just means being negative and being able to detect either the IgM or IgG, in this case, antibodies in a particular patient. So in order to combat these questions, the scientists came up with 3 different scenarios. So the first is what we call synchronous seroconversion. And so what that means is that you're able to detect both IgG and IgM antibodies at the same time post-symptoms onset. The next scenario is if a patient produces IgM earlier than IgG, and this is kind of the model that scientists use for other viral infections, where IgM is being detected first, followed by IgG antibodies a couple days after they're able to detect IgM. And the third seroconversion method is whether or not you can detect IgG antibodies earlier than you can detect IgM. So here you can detect IgG. A few days later, you can then detect IgM. So another poll. Which version of seroconversion do you think is occurring with patients who've been infected with COVID-19? Do you think it's synchronous seroconversion? Do you think it's IgM first and then IgG? Or do you think it's IgG first followed by IgM? I'll give you a couple seconds to answer. Awesome. So we'll end the poll. And so about 75% of the audience thinks that IgM are produced first, and then IgG, and then about 20% thinks it's IgM and IgG being produced or detected at the same time. Now, I'm not going to tell you the answer, I'm just going to show you the data. All right, so in order for the authors to look at this seroconversion that's happening, a couple of things that they looked at. So they have 63 patients and they're able to have serum samples in 3-day intervals. Now, out of those 63 patients, 96% of them were able to seroconvert antibodies. So they were able to detect either IgG, IgM, or both of these antibodies. But within that 63 patients, they were able to gather 27 patients. When they were hospitalized, they were initially seronegative. So what seronegative means is that they are unable to detect the presence of IgG or IgM antibodies. And so they took this sample and these samples of patients, and they looked at when these patients are producing either IgM or IgG. All right, so let's see what the scientists find out. So now we're looking at individual data sets. So each dot, again, is looking at each individual person producing a certain type of antibodies. So let's zone in on the x-axis. So here we're looking at the days after symptoms onset. And on the y-axis, we're just looking at the amount of either the IgG or the IgM. The red circles are IgG and the blue triangles are IgM. And so for 10 out of those 27 patients, they're actually seeing that they're able to detect IgG before they're able to detect IgM. And the median days of seroconversion where they can detect these antibodies is about 2 weeks or 13 days post symptoms onset. And so what this will look like is something like this, where you're seeing or you're being able to detect IgG antibodies in comparison to IgM antibodies, which happens a short time after. Next, they were able to show that 9 out of those 27 patients actually produced IgM and IgG antibodies at the same time. And so on the bottom, the x-axis is the same. So you're seeing about 6 days after symptoms onset, you have patients where you can detect both of these antibodies. Once again, seroconversion of both of these antibodies occur on average about 13 days. And here's what that graph would look like, where you have the detectable levels of IgM and IgG antibodies occurring at the same time. And then the patients, the scientists had 7 of those 27 patients actually produced or seroconverted IgM antibodies before they produced IgG antibodies. Once again, the x-axis is the days after the patient started to have symptoms, where you're able to detect IgM and antibodies, and later you can detect the IgG antibodies, with 13 days being that median day of seroconversion. So this graph would look something like this, where you're producing IgM antibodies prior to IgG antibodies. So the answer to the question is that it happens differently in different patients. I think this is another reason to point out sometimes how individual data sets actually is super important when we're looking at approaches for medicines. I'm gonna move on to the second goal. And so I've shown you within the sample set that the scientists have when they're producing the antibodies and how these antibodies are seroconverted within these patients. But now that authors want to ask, how can serology testing help identify COVID-19? Now, serology testing is just a fancy way to say that we're testing for the presence of antibodies. And so I just fine-tuned the graph based on the data that I gave you. Remember, this is not really a one for all, but this is just a depiction of like the aggregate data set. And so on the bottom, we have the days post symptoms onset. And so what you'll see in the beginning, when you have this viral infection, you can test with RT-PCR. And you can see if you have levels of the virus within that patient to show that they're currently infected with COVID-19. Now, a lot of times patients only go get tested if they're having symptoms. So you're feeling crummy one day, the symptoms occur, you're like, oh crap, this might be COVID-19. You get the viral test, they tell you that you're positive. But I just want to remind you that there are patients who actually never have symptoms, right? The only times until recently where patients would get tested is if they've been into close contact with patients who had confirmed COVID-19. So you have the asymptomatic patients that may or may not get tested. Now, if the test is super accurate, you'll see that they tested positive for the presence of the virus in their body by using the RT-PCR test. But there are cases where you can get false negative results where the test can tell you that you're negative for the virus, but maybe in actuality, you've actually contracted COVID-19 and you were asymptomatic and you did not know that you had the presence of that virus in your body. Now, the serological test is where we can look for IgM and IgG, and looking at the presence of these antibodies can allow scientists to figure out if a patient have the presence of IgM, meaning that they have been infected with COVID-19, or the presence of IgG, meaning that they are currently infected or they've recovered from COVID-19 infection. And so what these authors did is look at a number of patients who have come into close contact with those who have confirmed COVID-19. And so they have 164 of those patients, and they took those patients and they used the RT-PCR test to look for the virus. And out of those 164, 16 of them tested positive using the RT-PCR test. Out of those 16, 13 of them were symptomatic, so they had symptoms, and 3 of them were asymptomatic, and all of them tested positive for IgG or IgM or both. Now, the scientists also did RT-PCR tests on the rest of the patients, and these patients had no symptoms, and they also tested negative by the RT-PCR test. Now, 30 days after they tested negative, they gave all the patients an antibody test, and to their surprise, they were able to see that 7 of them, they were able to detect IgG and or IgM antibodies to SARS-CoV-2. And so what this is saying is that not only did we have 10 patients out of the 164 close contacts that were asymptomatic, but the RT-PCR missed about 4% of the patients who actually did contract COVID-19. So what lessons can we get from this, and what story does this tell? And so it appears that scientists are looking to initiate a way that we can combine antibody testing with RT-PCR tests, because it may be critical to identify patients who currently have COVID-19 and those who've recovered from infection. And so if we're able to complement the two testings, this would allow us to increase the accuracy for infection. And so they also showed that all of the patients that they looked at that were infected with COVID-19 produced or seroconverted detectable levels of IgG. Now, this IgG antibody, like I mentioned before, is the antibody that scientists think may infer immunity, but we're still unsure as to how long this immunity lasts. And if you wanted to learn more about if you can get reinfected, I would like if you can check out Joe's talk, who talked about a study that scientists did in macaques, where they were unable to reinfect these monkeys with SARS-CoV-2, but also you want to bear in mind that this may not necessarily translate to humans. And they also showed that patients produce IgM and IgG antibodies that can be detected at different time points after they started having symptoms. But on average, if we're looking at the aggregate data, it appears to be about 2 weeks after you start having symptoms to COVID-19. And so I think this is great that the study is telling us all of this, but once again, with every scientific article and every experiment, there are some limitations. And so one, they did not test for the presence of neutralizing antibodies. What are neutralizing antibodies, you ask? And so these antibodies basically have the ability to bind that specific virus, code it so that it's unable to infect the cells. So it gets rid of the virus even before it comes into contact with your cells. Even though we're producing antibodies, the antibodies may also be necessary for recruiting other cells or other molecules to get rid of infection, but infection may already be taking place. A new paper just came out in bioRxiv. So this is a preprint. It came out of Rockefeller University, my employer. And so what they're showing is that the patients that they looked at, so these are all patients in New York City. This data set that I'm showing you today are all patients from China. And from the data set that they're looking at, it appears that only a few people who've been infected with SARS-CoV-2 are producing high amounts of neutralizing antibodies, but it doesn't mean that the others are not producing any at all. It just might be a minute amount of antibodies that they're unable to detect in the laboratory. So feel free to check out that paper. Also, I just want to pinpoint that the sample size of this study is fairly small. So we only look at about 280 patients for a lot of the data that I showed you. But even when we're looking at severe versus critical patients, there are only about 20 that we classified as severe and critical. And so even though it showed a trend with the aggregate data, we're still unsure as to how the antibody correlates with the clinical course of infection. And also, this does not look at the changes in antibody responses for a prolonged period of time. Remember, the scientists only had the ability to test patients who were hospitalized. And so once they were able to leave the hospital, they're unable to test them. So even though we are producing IgG antibodies, and they also showed other data where there seemed to be like the IgG antibodies are plateauing, we can't really say anything for past about 25 days post symptoms onset because the authors did not look into that.